Yeah, our problem and our challenge is trust. And I think this is going to permeate everything we hear today. And to start that all off, our first speaker this morning is Jeff Glenn. Jeff is the Joseph Grant Professor of Medicine and Microbiology and Immunology at Stanford. He's the director of the Stanford Biosecurity and Pandemic Preparedness Initiative. He served on the FDA Antiviral Advisory Committee, and currently he heads Stanford's Antiviral Drug Discovery Center. So, Jeff, it's a pleasure having you with us.
Thank you very much, Lucy, and it's a real great to be here and participate in this, what looks like an extremely exciting symposium today. Before I begin, just for purposes of full disclosure, I want to say I've been a consultant to, have received research funding from, or have an equity interest in these entities. So viruses have been with us for a long time. We see evidence of smallpox and mummies from Egypt, atrophied limbs suggesting from polio infection. And of course, as Lucy was alluding to, in today's time, with travel and migration, immigration, faraway viruses come to our shores very quickly. And then this is how I used to always start my talks, and then now I kind of also think about BC and AC, which is before COVID and after COVID, because it really changed how we think about things. And that actually, one of the things that it led me to do was to initiate the Stanford Biosecurity and Pandemic Preparedness Initiative, or VIREX at Stanford, with a mission really to build out our collective antiviral toolkit proactively rather than reactively. Here's the website if you want to go visit it. And really reflecting on the comments that Lloyd was saying, it was a time when everyone came together and you realize how many people on this campus are working on things highly relevant to this mission, and in terms of developing inhibitors of RNA viruses, DNA viruses, modulators of the immune response, new technologies, detection methods, and everyone I approached said, yes, let's be part of this effort. And it really leverages and bridges across a lot of the key infrastructure we have here and stakeholders at Stanford. And we configured a couple of scientific cores to support this. One, a structural biology core with cryo-EM, pandemic assistance core, which provided us BSL-3 and even BSL-4 testing in animals and even in people if needed. And what we called the translation accelerator core to provide all the resources and bring in the expertise from industry that could really advise and make sure our science is really moving to deliver real products and cover all aspects from chemistry, process chemistry, regulatory toxicology, et cetera. And then Innovation Cauldron, which is an idea to really support new projects and provide training for people interested in this area. And because pandemics are global, so are we, so we've been establishing hubs around the world. First one was VIREX at Stanford, Vietnam, initially for tropical diseases. And here the co-director, Dr. Edward Pham, we're meeting with the Prime Minister of Vietnam. And I can vouch that Ruth O'Hara, who you can see in the background, it was a fantastic diplomat. And you and Ashley also joined us there. We also hosted the president of Vietnam when he was out here for APEC. It was the first visit of the head of state of Vietnam to Stanford. We have a VIREX at Stanford, Israel, which provides access to a great phase 1 clinical trial units, unique testing capabilities, supports some projects, and here Dr. Menashe Elazar is the co-director, seen here with the president. A VIREX at Stanford, Mongolia for liver diseases, and there we've done a lot of projects on surveys of different forms of hepatitis, liver cancer, and performed several clinical trials there. And here we're seen with the co-director, Dr. Naran Dashtorg, and with the Speaker of the Parliament. And finally, a VIREX at Stanford, Brazil, where we have access to a great phase 2-3 clinical trial infrastructure. And that's where we did our phase 3 study of a single dose of interferon lambda for COVID. And it really, to date, I still think it's the most effective drug that we have against COVID. It really this was the first study in a largely vaccinated population. And even in that population, there was a 51% decrease of hospitalization. And among those patients who were not vaccinated and treated early, there was an 89% decrease in COVID-related hospitalizations or death, which is the same number as Paxlovid, but there's no rebound. 100% compliance, because it's one dose and you're done, works across all variants, and the side effects were the same as placebo. Importantly, Lambda is the body's first line of defense, so it could be very good for other viruses that may show up. So, we can develop new therapies here at Stanford. They can go into phase 1, Virx at Stanford, Israel, phase 2, 3, Virx at Stanford, Brazil, and it's a whole new ecosystem focused on developing drugs for pandemics. And then, as Lucy mentioned, the Biden administration put out an RFA for these AVID centers, antiviral drug discovery centers. It was $1 billion touted to support 8 centers anywhere in the world. At the end of the day, it was a little political, all in the United States. 3 on the East Coast, 3 on the West Coast, 3 in the middle. But I'm very excited to be leading the Stanford one, and it regroups 60-plus faculty, consultants, their lab members, and admins and staff, et cetera. And really our goals are to develop towards the clinic novel direct-acting antivirals that have different mechanisms of action so they can be used together for at least hopefully additivity, but even more synergistic antiviral activity. And it really was modeled and built on Virx at Stanford, so the same scientific cores, now supporting 7 different projects, each co-led by multiple faculty. And these target many different aspects of viral life cycles, from targeting viral genomes, new RNA therapeutics, targeting lipid envelopes of viruses, using targeted protein degradation, of course, inhibitors of the viral protease, the polymerase, and because nucleoside analogs, which is a classic way of targeting polymerases, are unfortunately, the challenge is that the virus of SARS-CoV-2 has an exonuclease, so it removes those. So that project also is focused on developing inhibitors of the exonuclease, and that hopefully could lead to better efficacy when used in combination with nucleoside analogs. And finally, project focused on inhibiting the membrane-associated replication of viruses. And just to give you a dashboard of where we are, I'm very proud. We have a variety of projects that are moving along that translational spectrum. Some have now entered IND-enabling studies. And I do believe our center will deliver things that will get to an IND, which was the original purpose of the AVID Center. Unfortunately, the future funding of it is in doubt at the moment, but we'll leverage what we've done to date. And so I thought I'd just give you a sample of 2 of the projects that I'm really excited to be leading within this consortium to give you a flavor of the antiviral strategies. One is developing therapeutics that target highly conserved RNA secondary structures in viruses, so-called programmable antivirals. And the idea here is we want to target specific aspects of the RNA genomes of viruses. RNA is typically diagrammed as a string, but it really doesn't exist like that. It folds on itself, it creates these stems, these loops. Those are called secondary structures. And they often encode critical regulatory signals, and such as packaging or transcription, and they're often highly conserved. So I figure if they're really important and they don't change much, then if we design drugs designed to bind and distort them, there could be limited degrees of freedom that the virus would have to evolve against the drug and still be viable, and that could translate to a high barrier to resistance. So we established a method of doing high-throughput, high-resolution mapping of RNA secondary structure, validated on the HCV genome, where there's some known secondary structures, and got the same as predicted from classic high-resolution methods, and then applied it to influenza, and discovered a novel secondary structure, which turns out to be a packaging signal, PSL2. And this packaging signal is conserved across all known influenza A isolates. Bird flu, 1918 pandemic, even H5N1. And so then, since we knew the structure, we could design a so-called some short locked nucleic acid antisense oligonucleotides to bind and distort different aspects of this packaging signal. These turn out to be extremely potent in vitro. And importantly, we could squirt one dose into the nose of a mouse before, day of, and day after a lethal inoculum of influenza was delivered, and got 100% survival. And because we incorporate these 2 design features, one targeting highly conserved secondary structures, and also making our LNAs as so-called gapmers, so LNA on both ends, but single-stranded DNA in the middle, So when it binds and distorts its target, it also creates an RNA-DNA hybrid that is now a substrate for RNase H that can then catalytically destroy the RNA target. Those together make our LNAs 1,000 times more potent than a regular antisense oligonucleotide randomly designed in the same general area. And they turn out, indeed, as originally hypothesized, to have a very high barrier to resistance. In fact, we've been unable to select for resistance. Here we can grow a virus in the presence for 10 generations under conditions designed to promote resistance, and the LNA is equally potent picomolar efficacy at the end as it was at the beginning. Whereas in parallel, we do the same experiment with Tamiflu. We get high-level resistance after just 7 generations. And the LNAs work equally well against wild-type and Tamiflu-resistant virus, so that's very nice. And importantly, our lead LNA, one of our most potent ones, we can squirt 1 dose now into the nose of a mouse 2 weeks before a lethal inoculum and get 100% survival. We can give 1 dose 3 days after a lethal inoculum when Tamiflu does nothing and get 100% survival. And not only that, but the mice become fully immune. We can come in 2 months later, give them 10 times the lethal dose, and they don't even bat an eye. So call this just-in-time universal vaccine for influenza. Of course, here we gave it as a drop with a pipette min into the nose of a mouse. That's not how we're going to give it in people. So I said, let's use the same formulation, same delivery device we'll use in the clinic. And there we're thinking of a intranasal aerosol device like the flu mist vaccine. But the only problem is that doesn't fit into the nose of a mouse or a rat or a ferret. It does fit in dogs, and it does fit in pigs. And pigs not only have big nostrils, but they are a model of influenza. So we could send some of our drug and device to a colleague at USDA, who's a world expert in swine influenza. Most importantly, she said it was super well tolerated. I always say you can fix efficacy, but toxicity is a much harder problem. And then a few weeks later, we got back to virology, very potent data. Antiviral efficacy. And then we did another study. We could do a natural infection where just expose pigs to other pigs that have already been infected and also could very efficiently prevent the infection. And then we say, okay, well, what about tolerability? So then we've delivered this at 10 to 20 times the estimated efficacious dose in pigs, and no adverse events. We've done this also in dogs. Either giving it multiple days in a row or multiple doses in a single day. And no clinical signs, no histopathology, no test article related to anything. So very well tolerated. We have a very nice therapeutic index there. We've manufactured this non-GMP and GMP, and here's our TPP to really develop a single dose therapeutic or preventative for pandemic influenza, of course also for seasonal influenza. And that's actually an important point because I think any effort at developing a drug for a pandemic, I think it's really important to have a sustainable business model because we don't know when the pandemic is going to show up, and we can talk more about that. But this strategy can be applied to any virus of interest, so we call these programmable antivirals, so we could really design or program these against any virus, and when the sequence of SARS-CoV-2 came along, it gave us the opportunity to really test that. And we could very quickly develop extremely potent LNAs against SARS-CoV-2 that can dramatically drop viral titers in the nose or in the lungs of Syrian hamsters here. And this is our path where we're shooting towards to take it into the clinic. For influenza, you can do human challenge studies. So we plan to do traditional SAD-MAD phase 1 in healthy volunteers, and then do a study where volunteers receive a standard inoculum virus, and we can give our drug in cohorts either before or after to demonstrate efficacy as a preventative or as a therapeutic. And so after that, then we can pursue 2 different pathways, one for prophylaxis indication, potential stockpiling, and one, so we could hopefully shut it down in Wuhan next time, and one to give after inoculation, use our Virix at Stanford Brazil network would be a great way to test it as a therapeutic. And again, that's the group where we were able to conduct this phase 3 study of Lambda for COVID. And so the next stage, and I'm actually excited, this is the first company to be spun out of Virix at Stanford, because that is the goal at some point. We need to move these off campus for a variety of reasons, including the cost. This is really a company, 4J Therapeutics, designed to developed this programmable antiviral technology, but the first and lead program is this universal prevention or treatment of influenza. It's called 4J because the motto of my lab is to be happy and go for the jugular, do the key experiment, so we called it 4J. We've licensed the IP from Stanford, and a couple of months ago, we were very excited to be awarded a $314 million contract from BARDA to bring all the development through that human phase 1 challenge, as well as a few other things that potentially to do, and now doing the Series A. So let me give another example of a project now with small molecules. And here we're seeking to focus on the membrane-associated replication of RNA viruses and trying to develop oral small molecule inhibitors of, in this case, the SARS-CoV-2 NSP4 mediated membrane association of SARS-CoV-2 replication, as well as other viruses of pandemic potential. So all positive strand viruses replicate their genome in association with intracellular membranes. They either do it on pre-existing membrane structures or they create novel membrane structures. And we had actually studied this quite a while ago in the context of hepatitis C. And we had focused on the protein NS4B that really is central to establishing the HCV-specific membranous replication structures. And we discovered a lot of the biochemistry that was contained in a small amphipathic helical segment of the protein. And we could synthesize a peptide corresponding to that, add it to a solution of lipid vesicles of a small diameter, and it would cause massive aggregation of these vesicles, as we could see under electron microscopy. And it had a lot of features very similar to the membrane structures set up by HCV within infected cells. And so that we used this as an assay to screen for small molecule inhibitors that could prevent this lipid vesicle aggregation, and found some molecules that were quite potent at doing that. And they were very good inhibitors of HCV replication. At this time, there were better drugs that came out from Gilead for hepatitis C, which have revolutionized how we treat hepatitis C, so these went into the freezer. Until SARS-CoV-2 came up, and we realized that the predicted topology of the SARS-CoV NSP4 protein had a lot of features reminiscent of the HCV NS4B, and it also was implicated in establishing the membrane replication structures that SARS-CoV-2 uses. And so we took one of our derivative of one of our potent analogs against HCV and tested it against SARS-CoV-2. And we were very excited to see it actually had some decent antiviral activity in vitro. And then that led us to hypothesize, well, there is an equivalent of the HCV NS4B protein in SARS-CoV-2, and it was likely this NSP4 protein, and that there's an amphipathic helix in the NSP4 protein that's analogous to that amphipathic helix from the HCV NS4B, and it could promote lipid vesicle aggregation, and then our inhibitor, we hypothesized, could disrupt that process. So we synthesized a peptide corresponding to the amphipathic helix, added to a solution of lipid vesicles, and indeed it could cause a very nice lipid vesicle aggregation. And we could show that a very nice dose-dependent inhibition of this aggregation could be achieved with this STF3577 compound that correlated nicely with the antiviral activity. It was specific to this in that another amphipathic helix that causes lipid vesicle aggregation, but not related to this protein, the drug had no effect against that. And further validating the mechanism of action with some cryo-ET experiments where we could see in the top right here, in cyan, those are the membranous replication structures established by SARS-CoV-2, and in green are the virus particles made. And then this is untreated cells, but if we treat cells that are infected with the virus, as in D, now we largely don't see those cyan structures anymore, and instead we see a lot of these smaller vesicular structures, which we think are the precursors of those cyan replication structures, and it's abrogated by this small molecule. And importantly, was very excited to see that our compound was highly synergistic with protease inhibitors, either developed within our AVID or the one that's in the Pfizer's Paxlovid, and it was additive with respect to nucleoside analogs. But the magnitude of synergy was so strong, it was predicted to be synergistic in vivo. The compound had pretty good in vitro DMPK profile, and so we actually could show a dose-dependent inhibition in an animal model of SARS-CoV-2 with this compound as a monotherapy. Importantly, when we combine it with the protease component of Paxlovid, we could really demonstrate that indeed the synergy we observed in vitro does translate to very potent in vivo synergy. And very fond of this quote because the lab that did these experiments for us tested all the compounds for COVID, and he said this was the most potent antiviral effect they had seen with this combination. So, and then the compound was well tolerated in rats, very good exposure in tissues, and so we think it could be used as is for COVID. And one nice thing is that we could combine it with the protease inhibitor component of Paxlovid, and we don't need the ritonavir anymore, and that would remove a lot of the drug-drug interaction problems of Paxlovid. But we also thought that a focused medicinal chemistry effort could identify more potent analogs. So we think, there's a homolog, this NSP4 and the HCV-NS4B amphipathic helix are very similar, but they're not identical. So we think some compounds like 3577 can inhibit both, but we could also imagine compounds specific for the specific amino acids in one helix versus the other target that could be more potent against the virus. And indeed, we've now made derivatives that are significantly more potent than the original lead. And again, this 5913 is our current favorite. It has very good exposures and dramatic in vivo activity. This is 100 times more potent than the Pfizer protease inhibitor in the same animal model, and it still is highly synergistic with the protease inhibitor. So we think this really is something that could be a nice combination for not only SARS-CoV-2, but other viruses where these compounds can work. And then we've tested these molecules against other viruses, again, thinking that because there are homologs of this target in other viruses, so we have some very nice derivatives that are very potent against chikungunya virus, some against dengue virus. But importantly, we're most excited recently is we said, well, what about influenza? There is another amphipathic helix within influenza M2 protein, and we screened some of our novel collection of molecules against the lipid vesicle mediated aggregation that was mediated by this amphipathic helix, and found a compound 6472 that was quite potent. And importantly, this was equally potent when we tested against a virus, against wild type or Tamiflu resistant virus or baloxavir resistant virus, or even a virus that has the H5N1 M2 amphipathic helix. And excitingly, it's also very synergistic with both Tamiflu and baloxavir. And of course, we continuing the medicinal chemistry effort. Here we have a molecule 6675 that's even more potent and has very nice submicromolar antiviral activity, good exposure in mice when given orally, and most importantly, when we give it in vivo, after infecting mice with a lethal inoculum of influenza, we have 100% protection from death. So we think this could be a very interesting new class of drugs to develop for influenza. And just want to highlight that, hopefully get a sense that by targeting these selected amphipathic helices in different viruses, we have a whole new class of very potent antivirals that we are developing in parallel now for SARS-CoV-2, dengue, chikungunya, and of course, influenza. So I just want to conclude that the next pandemic is coming, and it's only a question of when, not if. This time we'll have no excuse for not preparing. And we really ignore the lessons of COVID, I think, at our own peril. And it's too late to start developing antivirals when the pandemic shows up. We need to be proactive so that they're available on hand to shut things down. As I mentioned, we can talk more about this. I think these antivirals should have a sustainable business model in the absence of a pandemic. That's going to best promote their availability for when they're needed. Stanford, of course, I believe is an ideal place to lead antiviral development efforts, and there's multiple classes of exciting agents, LNA, small molecules, but also protein therapeutics, antibody therapeutics. And this really is a collective, multidisciplinary global enterprise. And I just want to make sure to acknowledge all the great people that I get to work for in my lab currently and in the past, and our collaborators and our funders. Thank you.
I'm sure Jeff would be happy to answer some questions. Let me just start with one sort of sneaky question. What's critical in all of this is timeline. You have things in phase 1, you have interesting, exciting results for antivirals. We are not only dealing with COVID, which is here, and long COVID. But we're also dealing with H5N1 on the horizon, in which we have no idea of the timeline. What is your timeline from phase 1 of LNA, or the one you just told us about, with respect to what we can get into humans if and when this next pandemic happens?
Yeah, that's great. And actually, of course, I didn't highlight it, but what I fear most is knowing that my graduate student, she can make an influenza virus today that could kill a couple 100 million people. She could put a few point mutations in there that would make it completely resistant to the few crummy drugs we have. She won't because she's very nice. But we have other friends around the world where it's much cheaper than an atom bomb. You put one suicide cougher on a plane and you can literally bring down a society. We see, because it would be 10 to 20 or 50 times worse than COVID. And COVID brought many countries to the brink already. So that's what I worry about, and that's really the focus of my efforts. So I think if they will wait, or if the natural virus will wait a couple of years, we'll be fine. Because then we'll have an LNA, or we'll have the oral small molecule. But if it shows up next week, I think Lambda is the best drug for that. Because we know Lambda is very effective against influenza. And it's a single dose. It's can be used as a prophylactic, as a treatment. And it was originally designed, Bristol Myers bought it for $1 billion from Zymogenetics to treat hepatitis C. Did beautiful studies showing it was just as effective as the interferon alpha we used to use for hepatitis C, but way better tolerated. And so they were planning to manufacture it on a massive scale to treat hundreds of million people. So all that technology is there, it's ready to go. It just needs to flip the switch.
Yeah,
so that's what...
Who's going to flip the switch?
Well, so that gets into this idea that I think if whoever's going to develop an antiviral for a pandemic, it's expensive, whether it's the government, investors, philanthropy, and to develop a drug for a virus that you don't know when it's going to show up, that's a big ask. And so one way to make that easier is that if there's another indication that you can use it for. So if it's a broad-spectrum antiviral or something against influenza, if it works against seasonal influenza, that's fine. Another approach, which I didn't talk about, are so-called host-targeting antivirals. So they target something in us that the virus needs. One advantage, we predict a high barrier to resistance because the genetic locus that we're targeting is not in the virus. It can mutate all at once. It's in us. And we've actually shown that in our first antiviral that we've taken into phase 3 for host targeting. No evidence of resistance. Every patient, every time point that's been sequenced. There's a potential for broad spectrum, because in biology, when there's a successful solution for something, it's often copied. And so one drug for multiple bugs, broad spectrum. And again, the host target isn't just waiting for the virus to show up, it has a host function. And now you have a potent pharmacological manipulator of the host function. Many diseases in medicine is too much or too little of a host function. So you can have a whole non-viral indication with the same drug. And in fact, the drug that I mentioned that we've done a successful phase 3 for hepatitis delta virus, and we'll be filing the NDA, that same drug was approved a couple of years ago for a completely unrelated disease called progeria, which is disease of premature aging of kids. They look like they're 90, but they're 8, and this prolongs their life.
What you're actually saying is we have a lot of stuff pivoting, ready to go, but then the timeline is uncertain.
Yeah, you have to move them now, or they won't be ready when you need them.
That's right.
Thank you. It's great that there are more drugs in the offing for flu and other diseases. I was wondering if you could tell us what the mechanism of the longevity of the LNA effect was.
Yeah, that's really, I mean, in that case, I think the LNA is, I think about it as a very, very potent antiviral. So it's suppressing the infection dramatically, highly attenuated, but not completely. So it allows just enough low level of natural virus replication to occur so it can broadly stimulate the immune system, even better than a few denatured peptides in a vaccine. And we do see high levels of antibody produced against viral proteins, virus-specific T cells. And so I think that's how we view the mechanism. And it also would be a good partner to add with a vaccine, because a vaccine, assuming you have an effective vaccine, it takes time usually to ramp up. And so the LNA can give immediate protection and while that's happening.
So the longer-term protection is from immunity, not?
Yes, yes. I mean, there is some that's from the drug, right? We can give it, and then 2 weeks later still have protection, but 2 months later it's from the immunity.
Okay, so I just wanted to comment on something, which is that drugs, unless they stop transmission, which most of them don't, are not a pandemic solution. You know, so what they do is to ease the pathogen into endemicity. That's what happened with COVID, right? So what happens is that you develop the drugs, which is a great scientific work. We need the drugs, so let's have them. Then they are manufactured in small quantities. They are available for rich people. Rich people have the power. They feel insulated, and then they just completely wash their hands off the whole containment issue, and then everyone else can do whatever they want. So is anyone thinking about just making these things at scale, making them available to everyone, so that you don't have this effect?
Yeah, in fact, that is the mission of Virix at Stanford, and actually all the companies that I've started, is we think we have game-changing drugs, but we want them available to everybody at the same time at prices they can afford. We think that's best done through per capita GDP-dependent pricing with an effective anti-leakage program. So what we give to Mongolia stays in Mongolia. We will make money there, but we'll have huge impact. And we can make our money in the first world. But everyone benefits the same at the same time. And all these things can and are being envisaged to be manufactured at scale. But of course, if you manufacture them ahead of time, you may not need as many, because you can shut down, if they're available and stockpiled, you can shut down an epidemic before it really starts spreading.
Jeff, thanks. That was a mind-blowing talk. Incredible stuff. One thing that constrains a lot of licensing is a traditional model of licensing one drug at a time. And ultimately, if you look at hepatitis C or HIV or tuberculosis, you're going to actually combine drugs for their efficacy and sometimes to decrease their side effects. Since your drugs have such low toxicity, is there any possibility that they will modify the approval process so that you can approve multiple drugs at the same time? And I can say that one thing we saw with COVID that is appreciated is that the approval process for vaccines actually was modified. So I'm just wondering, what about multiple drugs tested at the same time for approval?
Yeah, that would be great. I mean, typically, I do think the antiviral division has been reasonably good in now, not in the past, but maybe, but of enabling early combinations. Typically, still want to see at least a couple of days of safety, with one versus the other, showing, but then you can go pretty quickly into combinations. And of course, for viruses, combination therapy is the name of the game, because you can decrease the emergence of resistance, you can increase efficacy. Of course, when we think about combining cocktails of antivirals for increased efficacy, we have to remember, we also bring in combinations they all bring in their own toxicity. So having drugs that are non-toxic that can be added to others is ideal, because then you can get the real benefit of combination therapy.
Although it also gives you the possibility of decreasing doses to actually
Absolutely.
decrease toxicity. So.
And in fact, the NSP4 inhibitors with the protease inhibitors is a great example of that, where we don't need the ritonavir anymore, and we can give them at lower doses, and yet they're still highly effective because of the dramatic synergy. But synergy is usually rare, and usually most drugs you're lucky if they're additive.
Okay, thank you very
Great.
much.